Yeah, 2010. I hope we don't have another one of those in the near future. So quite a challenging year for everybody, particularly chickpea growers. What I plan to do today, we're just going to, I guess, cover some of the things that happened last year, why they happened, and also go into the consequences for this season and what we can do for this season to minimise the risk for people who are going to grow chickpeas. Okay, so here are the culprits. And we'd expect to get all these diseases in a wet year. And I guess the thing that was unexpected was how quickly they came in, how quickly they developed, particularly ascochyta and botrytis. And we also saw a lot of waterlogging, a lot of flower abortion and poor pod set. Now that was not necessarily due to diseases. We know that waterlogging can affect flowers and pod set. We also know cool weather can do it as well. So it was a combination of those things that certainly affected the crops. So waterlogging is a pretty obvious thing to see. Very large areas affected. Very difficult to tell whether it was waterlogging or Phytophthora just by looking at them. And even when plants were pulled up, it was quite difficult to tell. Phytophthora causes the rotting of the lateral root, and then later rotting of the taproot. But in severely waterlogged plants, that can happen as well down the track. So sclerotinia, we saw a lot more sclerotinia this year in crops than other years. There are two species of sclerotinia, Sclerotinia sclerodiorum and Sclerotinia minor. They can both attack the base of the plants. Sclerotinia sclerodiorum, the sclerotes that are produced, which are those little black bodies there, they're just hard fungal structures. They can survive for a long period of time. Under very humid conditions, they can germinate, produce a fruiting body. The spores are airborne, so you can get above ground infection. It's got a very wide host range. Most broadleaf weeds and crops can be affected by sclerotinia. Once you get sclerotinia in a paddock, it's there virtually forever. Very difficult to manage. So botrytis grey mould, that was one that really crept up on us later in the season. And crops often look fantastic when you drove past them, but as soon as you went in there, particularly late in the season, opened the canopy up, there was the grey mould all over the dead tissue. Now botrytis grey mould, as well as causing infections and causing lesions, it can also infect dead tissue and decaying tissue. So here's just some images of botrytis grey mould on stems and flowers. A lot of botrytis grey mould early on tended to occur low in the canopy. And on that lesion, you can get millions of spores being produced. They're very small, and you would have noticed when you went into crops that had a lot of botrytis, when you open it up, the spores just took off and flew around. Very small, very light, you can travel long distances. So I'll have a look at some of the contributing factors to the 2010 epidemic, or epidemics. The tendency, I guess, towards short chickpea rotation. So instead of chickpeas once every 3 or 4 years, there's been a tendency, I guess, to narrow that back a bit. And what effect that has is it builds up levels of inoculum. Early planting and narrow rows. There's a big tendency for both those. And the issue there really is that, certainly in this season, with a combination of both of those, early canopy closure, very difficult to get fungicides in there, and also high levels of humidity in the canopy, very difficult to control. The first or second fungicide spray for ascochyta is critical. Certainly if the first one's missed and there's high levels of inoculum, very difficult to get under control, finally. Frequent overcast showery weather, and I guess it contributed to two things. One is vegetative growth of chickpeas. but it also meant there were repeated cycles of infection. And both Ascochyta botrytis, we're looking at a 7 to 10 day turnaround in the cycle, so things can build up very rapidly. And we're all aware of the issues with supply of effective chemicals, which resulted in some emergency permits being applied for. So these next two slides really just indicate what we all know. So this is a monthly rainfall for 2010 versus long-term average, and quite significant increases in rainfall, particularly over July, August and September. Probably more critical with a number of rain days. Once again, July, August, September, double really the number of rain days, certainly Goondiwindi, than usual. So double the number of potential infection events. We'll talk a little bit about the biology of these pathogens. You've really got to understand the biology to understand how they can be managed. So we look at Ascochyta. So it's really the main methods of survival is stubble, seed and chickpea volunteers. And really all those have to be managed to get effective Ascochyta control, as well as the other things we need to do like fungicides. So we look at botrytis. It's really stubble and seed. And as I mentioned before, botrytis cinerea has got a massive host range. Fava beans is one of the hosts of botrytis cinerea. For Phytophthora produces resistant spores that can survive in the soil for very long periods of time, years in fact. And with Sclerotinia, we've got these sclerotes, these hard sclerotes. And we're talking the order of probably 10 years or more, but the numbers really never really die out completely once you get Sclerotinia. So look at the modes of spread. Once again, stubble seed, very important. Within a crop, it's spores that are splashed from the fruiting bodies up onto the canopy and to adjacent plants. For botrytis, seed-borne infection is a significant mode of spread. So if a seed's infected with botrytis and it's not treated with a fungicide, it can cause seedling damping off or seedling death, which can significantly affect your population. As I mentioned before, airborne spores. So airborne spores can float around tens of kilometres or even hundreds of kilometres under the right conditions. And with Phytophthora and Sclerotinia, it's really mostly about soil movement and water movement of those resistant oospores and sclerotes. This is just a little bit of a hypothetical, but it really indicates how rapidly the spore numbers can increase over a relatively short period of time. So we're talking about a 7 to 10 day infection cycle. So you start with one spore, infects the stem, produces a lesion, then produces spores. And we've done some measurements, like I said before, and in a couple of centimetres leaf lesion, you'll have millions of spores. So you can see from that how rapidly diseases like botrytis can build up. So what are the consequences for 2011? There are low supplies of disease-free seed, higher levels of infected residues, particularly for Ascochyta and botrytis grey mould, higher levels of soil-borne inoculum of Phytophthora and Sclerotinia, where that's been a problem. And all that adds up to crops in 2011 being at a higher risk from these diseases than they are in normal years. And I guess it's all about integrating a whole lot of different disease management strategies to minimise it. And you've really got to start, right from the start. Last year there was a lot of flooding, so there's a lot of movement of stubble and infected stubble. So I guess the the rule of thumb about ascochyta, about not growing certainly, or growing it, at least 500 metres away from another, from a chickpea paddock from last year, isn't going to be as effective in a lot of cases because of that inoculum that's been moved around. But at the very least, we're advising don't plant back into paddocks where diseases were a problem. I mean, it's sort of a given really. Don't plant chickpeas in a badly draining paddocks. Kevin will talk a little bit later on about the relationship between waterlogging and other diseases. Controlling volunteers and weeds. So that's controlling volunteers, particularly for ascochyta, is really, really important. So we look at varieties. PBA hat-trick performed pretty well last year with ascochyta. It's got pretty good levels of resistance to Phytophthora as well. But no varieties have resistance to botrytis or to sclerotinia. So we've got to rely on other management practices to control botrytis. So we look at seed. Planting good quality seed is going to be really important this year. So it needs to have good germination, good vigour, low levels of asco and botrytis. If there's 2009 seed, you can access that, and that should be pretty good sort of seed. And also use a registered seed dressing. Once again, Kevin's going to talk about some of the trials he's done with seed dressings, so it'll reinforce that concept. And I mentioned before about earlier plantings, and I think you need to give a lot of consideration to planting a little bit later in the window to reduce that crop biomass. And also think about using wider rows. What that is going to do is to, I guess, push the canopy closure a little bit later. So, and hopefully maybe even having no canopy closure. So it's all about reducing the levels of humidity in the canopy, and it also means that you can get fungicides down there a lot more effectively. So we'll just turn to fungicides. There's no fungicides registered for sclerotinia or for Phytophthora. There's a seed dressing registered for Phytophthora, metalaxyl, but it's quite expensive. It only lasts for like 3 or 4 weeks after emergence, and after that, no protection. So this year, we're recommending that a fungicide, a preventative spray of fungicide, either mancozeb or carbendazim. Mancozeb is probably the preferred product. It's effective against ascochyta as well, whereas carbendazim's relatively, it's registered. Of the two of them, I think mancozeb would do a good job for BGM. So we're saying use a preventative spray before canopy closure. And also consider another spray 10 to 14 days later. For ascochyta, in a season like this where we expect to have high levels of pressure, we're recommending a preventative spray to all varieties, including hat-trick, and also a second spray later on. And what happens after that really depends on the weather conditions, how things go, and it's about following the current varietal recommendations. And they're all laid out in some of the brochures that you've got in your handout, so I'm not going to talk about them in any detail. I might hand it over to Kevin, and Kevin's going to talk about some of his seed dressing trials and the ascochyta fungicide trials.
Thanks, Mal. Good morning. Before we move on to the fungicide trials and the seed treatment trials, let's just revisit a couple of the things that Mal's highlighted. The first is, last year favoured huge increases in inoculum of all 4 pathogens. The second that he mentioned, surface water flow and floodings moved that inoculum out of the paddock in which it was produced across wide areas of the northern GRDC region. So what does this mean for you people? For your 2011 chickpea crops, it means this. These things are already in the paddocks that you're going to put chickpeas into this year, aren't they, Gav? Right? So down the Terry High Road, out at Mungandai, up at Weemallah, they're already there. When you plant chickpeas into a paddock that had wheat in it last year, you're planting into ascochyta. So if you only get one thing out of this morning, it is this. You must get those early ascochyta sprays out, otherwise your crops are going to get nailed with the first post-emergent rainfall event, and that includes hat trick. Righto. So the key to establishing a profitable chickpea crop in 2011 starts with this stuff. If you have healthy, viable, vigorous seed, you're doing the best thing possible to ensure a profitable chickpea crop. This is seed that was grown in 2009. This is 2 days after it's been wrapped up in a paper towel. Bill Yates set this up last Saturday. He's got 90% germination of his hat trick that he grew last year, and that's pretty good. So this is the sort of seed that we would like to be planting this year. Rapid germination, nice healthy radicals, 100% germination. Unfortunately, there's not a lot of that around. Most of the seed samples that we've been getting in through the lab at Tamworth look more like this than they do that. So this is 2010 crop, 5 days after we set it up. Not much germination, the radicals are smaller, and a lot of that seed's mouldy and will never germinate. So would you treat that seed and plant it? No, you would not. So we're suggesting if you've got seed that falls into this category, then you consider buying seed from a commercial seed supplier, because that seed will be germ and vigour tested, and if you ask them for the certificate, by law they have to give it to you. So we've been testing seed samples at Tamworth, and this is germination in that paper towel, and you can see it ranges from about 24% right up to 99% germination in paper towel. The red ones are 2 seed lots that Gordo sent to Tamworth last December. There's a DESI here, which I think is Kyabra based on the seed shape and size. 22% of that seed had botrytis, and this hat trick seed lot, 14% of that had botrytis. We didn't get ascochyta out of either of them. So we pursued those 2 seed samples, and we did to them what you people should be doing this year to your seed if you're going to keep it. You treat it. And you'll be treating it with either Thiram or pea pickle tea, which is what we did in this experiment. And we also had untreated. So we've got 3 treatments: no fungicide seed treatment, Thiram and pea pickle tea for the 2 seed lots. The yellow bars are the germinations after 2 days and the blue after 5 days. So the main messages here are not much difference in germination after 5 days, no difference amongst the seed treatments, about 80% for the DESI line and about 60% for the hat trick line. The hat trick wasn't quite as vigorous, it was a bit slower to get going. But again, 80% germination for the DESI, 60% for the hat trick, no difference between seed treatment. We then put that seed into, in this case, sand, in these little paper, plastic cups, and 10 days later I scored them for their emergence status. This one we called healthy because that one we thought would make it through to harvest. These 2 were abnormal and I didn't think they'd get through to harvest. So the blue bars, so here's our untreated DESI, our Thiram DESI and our pea pickle tea DESI. The blue bars is total emergence, the number of seedlings that came up in those little plastic cups. You'll see for this DESI line, seed treatments increase the total emergence. But that's not the true picture. The true picture's in the red bars. The red bars are the ones that I deemed wouldn't make it through to harvest. And you can see with both seed lots, treating the seed with either Thiram or pea pickle tea has increased the percentage of seedlings that wouldn't make it through to harvest. Which raises the question, should you be treating your seed? That's a question, guys. Someone want to proffer an answer? Jim? It would be an excellent idea to treat it. You would treat it. You'd treat it and you'd wear that loss right, from abnormals, because if you don't treat it, you'll end up with large patches in your crop, either from what Mal said, seedling disease caused by botrytis, or soil-borne disease. These fungicide seed treatments control both seed-borne diseases and soil-borne diseases. Treat your seed. So, if you want to keep your own seed, those 2 red samples that Gordo sent us, based on those, germination in paper towels is a poor predictor. Is a poor predictor of emergence in soil. So we're saying on that basis, you put some of your chickpea seed that you want to keep, you put it in the paddock now. You plant it in the paddock that you're going to put chickpeas in this year, and you see how much of it comes up. So if you plant 100 seeds, you count after a week and then after 2 weeks. If you need to water it up with a watering can, get it out, you do. And that will tell you if the seed's any good. It'll tell you whether the seed's worth keeping, right? Well, more importantly, it'll tell you if it's not worth keeping. So you plant 100 seeds, you get 20 out. What would you do, Peter Birch? You would. Wow. Wise move. You go to B&W and Moree, where the 2 big silos are. So we think that's a really important thing this year. Do this emergence test in your paddock. In most seasons, the germination in the paper towel is a good predictor of what will happen in the paddock. This year, based on the little bit of work we've done, that's not the case. And there are very good reasons for that. And we treat all our seed. So, very quickly, Gordo. Last year, Mal's already mentioned this, hat trick was superb against Ascochyta. It grew so well, unfortunately, that it fell over, and as Mal pointed out, it has no resistance to botrytis grey mould. So, unfortunately, although hat trick performed extremely well against Asco, in the end, the botrytis got it. Flipper didn't perform as well as we expected to because it got waterlogged, and in fact some Yorker crops had less Ascochyta than nearby Flipper crops, and they were all better than Jimbor and Kyabra. Where hat trick was planted next to or adjoining Jimbor or Kyabra, in which Ascochyta had not been managed, there was more Asco in the hat trick. And waterlogged crops had more disease, and in fact, waterlogging rendered disease management more difficult, which we'll talk about soon. So this is down at Balata. The bulk of the paddock was planted with rows running east-west to hat trick. The headlands, for some reason, the grower put Jimbor in his headlands, and as you can see, they were running north-south. They got hammered by Ascochyta in spite of the fact that this paddock had been sprayed with an effective fungicide. And as you can see on the 23rd of September, the Ascochytas killed the Jimbor plants. There's plenty of Ascochyta on these hat trick plants, but they're not dead. Right, coming back to this waterlogging thing, this is part of our Ascochyta management, one of our manager trials at Tamworth last year. This plot and that plot are both Kyabra. They both had 8 applications of 1 litre of chlorothalonil. This plot happened to be in a part of the trial that was severely waterlogged. And whilst most of what you're seeing there is a direct effect of waterlogging, there's more Ascochyta in that plot than in this plot, even though they both had 8 applications of Unite. So what's happening here is that waterlogging is interfering with our ability to manage Ascochyta using a strategy that has normally worked. So if you had problems last year in managing Asco, this may be the reason. Right, looking at the yields of that trial, so we've got 5 varieties, Kyabra, Jimbor, hat trick, and 2 numbered lines coming through the program. So this is a trial where we deliberately introduced Ascochyta when the plants were at the 3-leaf stage, and we had 3 fungicide treatments. A low Ascochyta treatment, the red plots, where we sprayed those plots 14 times with 1 litre of Unite 720. And the first spray went out before we inoculated the trial. So those red ones we always had the chemical out before the infection occurred. The yellow plots, they didn't get any fungicide. The blue ones, the variety management package, these two ones here, they got their first VMP spray the morning that we inoculated. So I sprayed those blue plots the morning before inoculating, about 2 hours before inoculating. So those blue plots were like the red plots for Kyabra and Jimbor, but at a lower rate, at an off-label rate. So the first thing, in susceptible varieties like Kyabra and Jimbor, we can manage Ascochyta with the right chemical and the right rate. So, you know, almost 3 tonnes per hectare in Jimbor last year. Where we didn't spray the Jimbor and the Kyabra, right, no yellow bars there, that's because we didn't put the header in them. The unprotected Jimbor and Kyabra were dead by the end of July, 6 weeks after we inoculated. The unprotected hat trick went 1750 kilos per hectare, and these new varieties went over 2 tonnes. No sprays. In a trial where we killed Jimbor and Kyabra in 6 weeks. These things had no sprays. We didn't harvest until the 23rd of December, and they still went 2 to 2.5 tonne per hectare. Now I must say this, we didn't have botrytis in this trial. So severe was the Ascochyta early on that all these varieties got infected during winter, and it slowed them down, it reduced their biomass, so we didn't have a problem with BGM. So this data is not representative of what happened in your paddock, but it's also not confounded by botrytis. So if you want to grow Kyabra and Jimbor in 2011 and make money out of them, make sure you get that first spray out and the second, and if it's a season like last year, you need 14 in the shed. If you're going to grow a hat trick, and you choose not to spray it at all, you should still get 1750 kilos per hectare. But if you want to make a bit of money out of hat trick, maximise your profit, you'll use this low and off-label rate if you live south of the river. Because where you guys are, you can't do it legally. Righto? You cannot use that low off-label rate in Queensland. We can use it in New South Wales, we can even recommend you use it in New South Wales, but we can't up here, and that's why I'm not telling you what the rate was. Who's thinking about growing chickpeas this year? Right, make sure that you understand that more than any other time in the history of chickpeas in this country, your 2011 chickpea crops will be at the highest risk of diseases ever. And if you're not prepared to understand that or admit it, don't plant chickpeas this year. We've already addressed the issue of seed. If you're confident and competent in managing Ascochyta, you can grow Jimbor and Kyabra and put your orders in with Birchie for your chemical pretty soon, because there'll be a run on it. Otherwise, grow only hat trick or varieties with Ascochyta ratings as good as or better. Mal's already mentioned about delaying sowing, and Jeremy will talk about sowing next up. These Ascochyta sprays. Down just north of the Plant Breeding Institute at Narrabri, on the right hand, on the eastern side of the road where there was some Albus lupins there last year, flipper chickpeas. Old mate missed the first spray across the whole paddock, and he missed the second spray across half the paddock. On the 18th of August, which was the second day of AgQuip, the part where he missed both sprays was dead. And the part where he missed only one spray died about a month later. That's flipper. Now, there was a waterlogging issue there, but flipper's normally considered to have very useful resistance to Ascochyta. Do not miss those sprays on any variety this year. And these other ones, well, the season will determine whether we're going to have a problem with those 3, Sclerophytophthora or botrytis.
Thanks. Yeah, just on the first 2 sprays with the chickpea, what product do you recommend, Mancozeb or chlorthalonil?
They're equally effective as protectants, yeah.
Well, what rate for Mancozeb, for example, do you recommend?
Kilo's all right for the first spray, yeah. But it's got to go out before the first post-emergent rainfall event. So it's a true protectant. You're not trying to salvage, you're trying to protect.
Kevin, if we are going to work with farm seed, say there's a lack of seed or lack of quality seed around, sure. We did talk about this earlier, whether by screening it, by putting it deep, would that be a better approach?
Planting deep, that'll sort out the dodgy stuff, it won't come up.
Yeah. But if you want to get seed off that crop and have better quality seed for the next year, would it be a strategy?
Well, there's two problems there, Jim. If you plant weather damaged seed and you plant it deep, you're going to end up with, unless you adjust your sowing rate, and you won't know how to do that because you won't know what percentage of that weathered seed will actually emerge. But let's say you put it down 6 inches, I would imagine you could only, the best you could hope for is 50% emergence. So you're going to have, you've either got to double your sowing rate, so if you're planting a hat trick at 80 kilos, you're looking at 180 kilos a hat trick, half of which isn't going to come up. And if it comes up patchy, you're looking at, you're running into a virus problem where you are. You know, we didn't see any viruses in chickpeas last year, but we know they've built up over summer and they could be a major problem this coming season.
If the amount of inoculum that is around should be burned. I'll start.
Is this for Ascochyta or other pathogens?
Sorry?
Is this, are you addressing Ascochyta or other pathogens? Well, I'll flick the botrytis and sclero to Mal, but from the Ascochyta point of view, what paddocks are you going to burn? All of them?
Yeah, that's a good point.
Because it's everywhere.
So it's the same with the botrytis as well, because I mentioned it's got a wide host range, you know, over 180 genera or 40 genera of plants or something, and it can colonise just about anything, any plant material that's dead. So, you know, doing that, and you're not going to get 100% burn anyway. So doing that might reduce it a little bit, but you've got all the stuff coming in from the outside. For sclero, I wouldn't have thought so. I wouldn't have thought burning would affect sclerotia too much. I mean, they usually fall in the soil and gradually work their way down. So,
yeah, as disease management tools, burning, bashing and burying are probably not going to do much. But from an agronomic point of view, they may be quite useful in terms of helping you get the crop in the ground.
What's the recommendation for CQ?
you know, source seed from CQ, and I know that's going to be difficult. I mean, we know that we've had outbreaks of Ascochyta up there 2008, 2009, 2010, earlier than that as well. But, you know, I think that's one of the keys to CQ, just trying to keep Ascochyta out. So that's the first thing. I think for, okay, we've had Ascochyta outbreaks there. So I think any crop that's been anywhere, is going to be grown anywhere near a known Ascochyta outbreak, it should be treated. It should be treated like varieties down here. Those first couple of preventative sprays. And I think the same for the botrytis, based on the higher levels of inoculum around.